Welcome back. We are starting chapter 42 and we are going to discuss biotechnology and industrial microbiology. We're only going to discuss the sections 42.1 through 42.3 in this chapter. Now, the subject of biotechnology and industrial biotechnology do have two parts. So one of the part is that we do exploit the microorganism to make something at the industrial scale for our benefit, the things that we need. Now, the first part, what we do is our knowledge of molecular biology, biochemistry, microbiology. These do help to isolate and characterize a microorganism from the environment. It could be a bacteria, it could be a fungus, many different types of microbes. And then we can either modify them by genetic manipulation, or we just identify a specific type of condition in which that particular organism will produce something that we want in a large amount. And this is the first idea of how we can use that microorganism in an industrial scale. So the first research starts at the laboratory level in a very small level. Once that particular organism is standardized in that process by genetic engineering, by biochemical manipulation, by mutation, or by identifying a very specific method or biochemical method or culturing condition in which that will produce a particular product, then the scale up goes on. And the scale up means that it is taken into the industrial level where the product can be produced in a very large amount and then subsequently purified, processed, and used for the human purposes. So it is a very multidisciplinary subject where it starts with some basic biochemistry, molecular biology, microbiology. Then it goes into a particular area which you call as an industrial microbiology or bioprocess engineering. And it really does need the clear understanding of engineering knowledge to scale up this process. So let's take a look at what we are going to talk about in this. So we are talking about that there are many products of the industrial importance that we use by these microorganisms. And let's take a look at what are these and how we can exploit these. So if you just look at these, that these are the major product of industrial microbiology, and all these products are produced by different microorganisms. So you can see that this is your baker's yeast, Saccharomyces cerevisiae, that can utilize glucose to produce ethanol. On the other hand, this species produce ethanol from the lactose, the milk sugar. On the other hand, acetone and butanol, I'm just giving a just example. Say, for example, your nail polish remover. Acetone is one of the constituents in there. It is a constituent of paints. You can find them in a hardware store. And this is industrially produced as well. Butanediol or butanol, these are some of the additives of many of the chemicals. These are produced by Atrobacter serratia. Many different types of enzymes that are industrial application, hydrolases, oxidases, hydrogenases. These are produced by a variety of microorganisms. On the other hand, you can see that the gibberellins, these are some of the plant hormones which can be produced by microorganisms as well. Food additives, there are a lot of them, lysine, glutamate that we use, that you can see that Corynebacterium glutamicum, this is used for the glutamate production. On the other hand, citric acid, you all know that citric acid, one of the organic acid, which is used as a food preservative, even in your home canning solutions. This is produced by microorganisms. Nucleotides, these are industrial applications. There are many different types of drugs that use this. On the other hand, vitamins, we just all know the importance of the vitamins, and these are produced by microorganisms. Polysaccharides. These polysaccharides are often used as food additives. There are many applications of that, even in the oil drilling, where these microorganism produced polysaccharides are needed as well. And we all talked about the antibiotic, which is a penicillium species, streptomyces, different bacillus. These are used for antibiotics. And from the initial product that is produced by the microorganism, you can always take them to modify those, which we call as a semi-synthetic antibiotics. Here is an interesting example. We talked about this Claviceps purpurea, which do produce an alkaloid, and it is an extremely toxic product. So on the other hand, this alkaloid is used specifically and tagged with some specific delivery molecule to kill cancer cells. And there are many different types of applications to that. On the other hand, many of the steroids that we use, even in our cream or even oral steroids, these can be produced by microorganisms. On the other hand, you can see a variety of hormones like insulin, human growth hormone, somatostatin, and interferons. Interferons is not a hormone. It is basically used for antiviral therapy. These are all produced by the microorganism. Now, I will come back to this because here you see that there is a word, recombinant DNA technology, that is not anywhere here. So what is the difference? And I will come back to that. And on the other hand, that hydrogen is now more and more being used to replace the traditional gas or gasoline that can be produced by the photosynthetic microorganism. Methane from this bacteria, and as well as ethanol can be produced by some of these bacteria as well. One of the ethanol that we are talking about mainly for the fermentation of alcoholic beverages, and then there are some of the ethanols that is used for biofuels that uses a different source, Zymomonas and Thermoanerobacter. These are mainly used to produce ethanol for biofuel. So most of these microorganisms that we are seeing, these are naturally obtained from the environment, and usually there are some naturally induced mutations that are taken care of, which mutations make them produce some specific amount of material more than the wild type of the original species. That's one of the things. Another thing is that you can manipulate the growing environment of many of these to get the specific product. Whereas here, that only for this that we talked about, recombinant DNA technology is entirely different. Here you take a particular organism, which is an E. coli or Saccharomyces cerevisiae, which is the yeast, and then what you do that you clone the gene of your interest, which will produce a protein, which could be insulin, human growth hormone, somatostatin, interferons, even viral vaccine components. You clone those genes in a plasmid and you introduce that plasmid into E. coli or the Saccharomyces yeast. You grow these organisms in a very large amount in the industrial setting, and these bacteria or the yeast will produce these, which are not the natural product of these, but these are cloned into a plasmid which is inside these, will produce these material like insulin growth hormones in a large amount. From the large growth in the industrial setting in large fermenters, you purify these and then you package those for human consumption. So very different things. Some of these things are the natural products which are obtained from the microorganisms, and we only have to manipulate the biochemical condition, or we have to choose a mutant which is capable of producing that particular material in a larger amount than the normal. And the second thing is that we genetically manipulate and introduce a particular gene which is not a natural product of that bacteria or the yeast or any of those microorganisms. This is used by the recombinant DNA technology. We make them produce the product that we want. So this is a very important concept. Just make sure that you understand the difference between the two products, natural product versus man-made product by recombinant DNA technology. So first, we are looking at the antibiotics, and more than 65% of all the antibiotics that are produced by microorganisms. And most of them are produced by the Streptomyces bacteria or filamentous fungi. For example, a filamentous fungi is penicillin right here. And think about that we are producing right here the penicillin. So to do that, you requires a precise control of nutrient. And why this is important? Because this maximizes the production of penicillin. So what is needed, if you just look at that at the initial condition, right here, you have this lactose level, and the lactose level or the fermentation of lactose is rapidly carried out. And as the lactose is going down, at certain extent, you can see a little bit of glucose is introduced into the media. So initially this glucose after a certain amount of growth, so you can see that this is the biomass. That means the penicillium is growing in logarithmic phase and then started to slow down. So after this logarithmic phase is done, you can see that it is fed with the glucose right here, not before that. After it passes the logarithmic phase of growth of biomass, it is fed with the glucose. This maximizes the production of the penicillin. Why it is so? If you feed the glucose right from the beginning, it will convert the glucose and produce its metabolites. And it is not a condition which is a stressed condition. What you are seeing that as the lactose in the media is getting down, at certain point after the lactose level is pretty low, the organism thinks that this is a stressed condition. And at the stressed condition, this penicillin will be produced much more by that organism, because think about that, what a penicillin or antibiotic is. The organism is trying to kill other organisms in the environment or in the vicinity, and it's trying to compete with them. When it will compete? It will compete when there is not enough nutrients in the environment. So here, that's the reason that when the glucose, if the glucose is available right from the beginning, then the organism thinks that, oh, there is plenty amount of food to eat and it is not in stressed condition. But here what we have, you have the lactose. Lactose is a disaccharide which is broken down into galactose and glucose. When the level is pretty low, at this point, the organism is trying to get stressed. You feed it with the glucose because if you don't, it was going to die. But you can see that first you initially try to produce 1.45 gram per liter. This is a little bit higher. Then a little less, little less. Why less and less? Because see that still when you have a little bit higher amount of glucose, it is still growing. And when it is getting into the saturation level, that means the biomass will not grow. You stress the organism even further by lowering the amount of glucose, so it produces the highest amount of penicillin. That's what you want. On the other hand, you can easily see that right here, you feed it with the nitrogen about 18 milligram per liter in this condition, which is after the log phase growth is done. You just add a little bit of nitrogen in there, which will enhance the production of the penicillin. And you can see right here, this is the amount of ammonia that is being produced by the microorganism. And right here, you can see that this is the production of penicillin, how it goes and maintained over the period of time. So this is how you maximize the production of the penicillin by manipulating the different conditions in the fermenter or where the penicillium is growing. Now, you can also stimulate the production of penicillin to something different. Say, for an example, if you just add phenylacetic acid into the media, it's going to convert the penicillin to penicillin G. And these are some of the modifications that you can do. It takes about a week to produce the penicillin. Once the penicillin is produced, you can absorb them, then you just purify them and then crystallize them, and then you can add or modify the side chain. Say, for an example, amoxicillin, ampicillin, these are some of the modifications of the native penicillin that we talked about in the antibiotic chapter, and that can be done after the purification process. So the next we are looking at some of the amino acids, and many of these amino acids are produced from regulatory mutants. And we're going to talk about that, what we mean by the regulatory mutants. And there are some like an lysine glutamic acid used in the food industry. You have heard about the monosodium glutamate. Some people use it, some people don't. Some food labels are there which says MSG free or glutamate free. We are not going to go into the detail, but again, these lysines are common in bread products. You will be able to see that. These are flavoring agents. So glutamate is produced by Corynebacterium glutamicum. And for the production of the glutamate, the mutant species can convert efficiently a specific step in the TCA or tricarboxylic acid cycle, which is also known as the Krebs cycle. So what happens that this step is the conversion of alpha ketoglutarate to succinyl-CoA. This particular bacteria, as it is limited in this particular step, what you do that you add fatty acid derivatives into the media, and also you keep the biotin level low. If it is a higher amount of biotin in that, it will try to produce succinyl-CoA from alpha ketoglutarate. but you limit the biotin in the media and you add the fatty acid. And what it does, that this increases the membrane permeability and secretion of high concentration of the glutamate into the media. Now, this impaired bacteria, which is a mutant bacteria, also use another cycle, which is known as a glyoxylate cycle. Why it has to use that? Because think about that. We talked about that it cannot convert efficiently from the alpha ketoglutarate stage to the succinyl-CoA. So if the bacteria cannot undergo the Krebs cycle or the TCA cycle, it's not going to produce the energy or the ATP that is needed for it to survive. So to survive, it needs to go or bypass that particular step so that it can produce the subsequent step of the Krebs cycle. And this is done, or the bypassing of that particular step is done by the glyoxylate cycle to meet their needs for the intermediate products for energy production. So what happens that by the time the nutrient is actually depleted and the growth stop, just by manipulating or not allowing that alpha ketoglutarate to succinyl-CoA conversion, there is almost a molar conversion of the isocitrate, which is the starting material, to the glutamate, which is the product that we actually want. And this is how just the regulatory mutants can produce a large amount of this industrial amino acids for use in the food industry. So next we are looking at the citric acid. And this is also the similar way, just like the previous one, we can manipulate some of these bacteria or the microorganism to produce organic acid. Citric acid, food preservative. Acetic acid is a vinegar. There are so many different types of vinegar, flavored vinegar. These are all industrially produced. Lactic acid, you just make your sauerkraut in the lactic acid. There are so many applications for these, you already know. The process used aspergillus, and what they do, that you limit the trace metals like manganese and iron to stop the growth of aspergillus at a specific point. So you take out these trace elements from the media, and that will not allow the growth of these aspergillus in the media. And generally what you do, that once that growth is done, you add very high sugar, like 15 to 18%, and this regulates the glycolysis followed by the TCA cycle. So if the sugar level is very high during the active growth phase, what happens, the citrate synthesis activity increases. That's what you want, to increase the citrate synthesis. On the other hand, the activity of the subsequent steps in the TCA cycle, you can just take a look just for your understanding that what's going on. We're not going to ask these in the exams or the quiz, but you want that citrate synthesis to produce this citric acid. I'm taking just one example. And then the subsequent steps, which is basically, there are two enzymes, which is aconitase, and then the other one is isocitrate dehydrogenase. You inhibit those two steps. So you want the step one, which is the citrate synthetase enzyme producing citric acid, but you want to inhibit the aconitase and the isocitrate dehydrogenase, which is going to convert the citric acid to the subsequent material. You do not want that. You want to stop the reaction of the TCA cycle at the citric acid. So this is basically the manipulation that is done by changing the environment of the fermentation condition in the media. And this can be done pretty easily. And you already know that most of the biochemical reactions are reversible. So there is an equilibrium constant when these reactions are going on. But by manipulating the conditions, you can make these reactions stop or not go forward at one point of time. And these are some of the essential process by which industrial microbiology works. So basically, by manipulating these steps, you produce the product that is desired in a large amount, and you do not want to form the product which will naturally form in a biochemical reaction in the organism. So the next one is a biopolymer, and used to modify the flow characteristic of liquid, serve as gelling agents, and there are many examples of that. So I'm just giving you some of the examples that what they are used. Say for an example, the dextrans are used for blood expander absorbance. This Erwinia polysaccharides are used in paints, surprisingly. The polyesters from Pseudomonas, and again, these Pseudomonas are not pathogens, of course. These are used in plastics, and these are biodegradable plastics that you see some of these bottles today which says biodegradable plastics, cellulose microfibrils. This is produced by an acetobacter, and this is used as a food thickener. Then there are some polysaccharides like scaleoglucan. These are used in the oil industry as drilling or mud additives. It's kind of interesting that why you want to do this, that when you drill, that thickens up the mud and that helps the isolation of the oil from that thickened mud much more easily. Another very interesting is the xanthan gums. You probably heard about this very commonly. Almost all food products will have that. So what is the use of these xanthan gums? You already know that many individuals are sensitive to gluten, and gluten is basically a binding agent which binds many of the food products. It's like a glue of food. So instead of gluten, you can use this xanthan gum to serve the safe purpose. And this helps the ingredients to stick to each other. And you will find this in your candies, in your chocolates, in your cakes, pastries, in breads, jam, jellies. And also even the xanthan gum is used in oil recovery from the thickening mud, just as this scaleoglucan polysaccharide. So these are some of the biopolymers, and these are biodegradable as well and environmentally friendly. Then is a biosurfactants. And think about that biosurfactants is kind of like a soap or detergent produced by the microorganism. And this increases the detergency, wetting, phase dispersion, solubilization of the material. And these are very important in bioremediation or oil spill dispersion, because oil and water doesn't mix. But the biosurfactants or the soap will mix the oil water and enhance either oil recovery or in the bioremediation that we're going to talk about in the next chapter. So what are the biosurfactants? So biosurfactants are nothing but it's an amphiphilic molecule. What we mean by an amphiphilic molecule, amphi means both. It does have two parts. So one part is called the hydrophilic part. Think about that. If you just go to your basic structure of your plasma membrane made up of phospholipid. So your phospholipid, the head group is made up of phosphate, negatively charged. That is your hydrophilic part, interacts with water. And then you have the fatty acid tails, which is hydrophobic and buried inside or sandwiched in between away from the water. So the same way, a biosurfactants molecule will have, say, a carbohydrate part, which is charged and interact with water. And then you will have a long fatty acid chain part, which is hydrophobic, and that is not happy to be with water. So what it does, the hydrophobic part will mix with oil or fat. I mean, remember that oil mixed with oil. The other part of the molecule, which is hydrophilic, will interact with the water, and that will help in the wetting, solubilization, phase dispersion, all these characteristics. So the next, what we are looking as a biocatalyst, and there are many enzymes like dehydrogenases, oxygenases, hydrolases, transferases, lyases, and all these enzymes are tremendously important in the industrial setting. And some of these enzymes may add a hydroxyl group. Some of them can add a keto group. They can saturate or desaturate a cyclic structure. Saturation means it will convert a double bond into a single bond by hydrogenation, or it will convert a single bond by taking out the hydrogen into a double bond. So these are some of the processes that is being used. Advantage of these, that using a microorganism rather than just a chemical process, that they produce the appropriate stereoisomer. What we mean by the stereoisomers, you all know that there are many products, say for example, amino acid, they are in the D and the L form. We always use the L form of the amino acid, not the D form. If you try to produce them just by chemical reaction, you produce these stereoisomer, which will be, many of them could be in the D form, and humans cannot use the D form of amino acid. On the other hand, the microorganisms are often very clever to produce the specific stereoisomers, whether it is D or L. And if it is L, that would be great for the human consumption. We can use that. So these are some of the advantage of that. They are very specific, and also you can use a mild condition to produce the products that you want compared to a harsh chemical reaction that you have to take. So here is a process that see what is been shown. This Rhizopus nigricans is producing a product. So let's take a look at what is actually being done. So here is a starting material, which is a derivative from cholesterol, and this is progesterone. So progesterone is a hormone as well, and this is the starting material for a lot of different products, and we'll come to that. So what you do, remember that this is only lipid soluble. So you first dissolve this in acetone, which is a nonpolar molecule, and the fat will completely dissolve in the acetone. And then you add that to the culture of growing Rhizopus nigricans, and this particular dissolved progesterone, which is the steroid, will make its way into the fungus. So once it goes inside the fungus or the Rhizopus, what it will do, that it will do a biochemical modification to add a hydroxyl group right here. And we call this as an 11-alpha hydroxylation. And why it is important, and it is written as a major product, this is not needed, but just an idea. This is an intermediate for your cortisone or hydroxycortisone. So you all know that your cortisone or hydroxycortisone is a main ingredient for your topical steroid. Over the counter that you find your itching creams, these are made up of your cortisone and hydroxycortisone. And this is the product which is the precursor with that. So these are some of the biocatalysts. This Rhizopus nigricans is using its biocatalysis power to convert this progesterone into this major industrial product from which many different products can be formed. So another very important thing is the vaccines. And these are critical weapons against infectious disease, which could be bacterial, fungal, and viral diseases. And sometimes traditional vaccine may be ineffective. And there are several ways that the vaccine can be produced, and we already talked about that somewhere else. So some of the processes that we already talked about, that you can isolate the microorganism and you can heat kill or chemically denature or chemically kill the microorganism. So heat killing or chemical killing of the microorganism can produce the dead microorganism that do have the same similar characteristic or surface characteristic of the proteins that you can inject for a vaccine. Many times that works very well. Sometimes some of these viral vaccines are live, but they are called attenuated vaccines, where these particular viruses are passage over the time and it loses its infectivity. So it can go into your cell, will just replicate very mildly for a short period of time. And within that short period of time, you have an immune response and you will raise antibody against them. And after that, your immune system is going to kill those remaining viruses. But you will have your memory T cells and memory B cells already being produced. So if you are challenged by the real virus in the future, you are protected with that. So, but here, what we are talking about, none of those. But sometimes some of these traditional vaccines may be ineffective. There are two ways. Sometimes you cannot grow a particular virus in large amount so that you can isolate and you can find an attenuated vaccine. Those are just simply not possible. So in those cases, what you do, what is called a reverse vaccinology, and we will just come to that the pathogen genome sequence to assemble a list of antigen that may be a possible good vaccine target. So to do that, what you are looking at, good antigen must be expressed by the pathogen during infection. It must be secreted on the surface of the pathogen, so it should be on the outside. That is the most important part. Be found in all strains of the pathogen, and that antigen should elicit an immune response. That means your immune system cells will be able to find it. And it is also essential for the pathogen survival. Having these idea about what is a good antigen, what you can do, that you can clone these peptides or proteins into a particular plasmid, and you can grow them artificially. So for an example, classic example for this is hepatitis B vaccine. Hepatitis B vaccine is produced in either E. coli or Saccharomyces cerevisiae, which is the baker's yeast. And this is a recombinant vaccine, which is basically a product of reverse vaccinology. So let's take a look at what is the real benefit of this reverse vaccinology. So, for an example, that there is a particular microorganism which is Neisseria meningitis, which do have five serovariants and or called the serovars, like A, B, C, D, E. Now, the vaccine against the A, then C, D, E was available for many years, but not for the serovar B, which is also called the Men B. So serogroup B. Why it is so? So all other four strains, which are the Neisseria meningitis A, C, D, E, the vaccine is against a polysaccharide that is found on the surface of this pathogen. But unfortunately, the interesting part is that the serogroup B do have the polysaccharide, which is very close to a human polysaccharide. So what the problem is that if you do have a vaccine against that, the immune cells would understand that serogroup B vaccines and will also attack the polysaccharides found on your own cells. And this will trigger an autoimmunity disorder. So you do not want that. So that was the reason that this particular serogroup B vaccine was not available for a long time. So the scientists started the reverse vaccinology. So what they did that they have the whole genomic data of the Neisseria meningitis, and they identified about more than 600 surface proteins. And once they identified these 600 surface proteins, they found out that maybe out of 300 could be a candidate for an antigen. And they tested about 300, half of them on the mice. And then from there, they just looked at that which are the ones which gave good amount of immune response, or the mice did produce antibody against them. And there were approximately 25 for the further study in the preclinical studies. So after that, preclinical studies or the clinical studies, phase 1, 2, 3 were done. And in 2014, the first vaccine, which is the Men B vaccine, was approved by the FDA for approval in the United States. There are many reverse vaccine is being developed for, say, example, Staphylococcus aureus strains, group B streptococcus, chlamydophila pneumoniae, which is causal for pneumonia, Bacillus anthracis. And interestingly, very relevant is that now a lot of different companies are racing against this COVID-19 virus, which is also known as the SARS coronavirus type 2. And most of these vaccine development are at the preclinical level. There is only one company which is based in Massachusetts, which is Moderna, that went into the phase 1 clinical trial. That means that there were 45 individuals that were injected by a vaccine. So this SARS coronavirus type 2 does have 4 different types of proteins, glycoproteins on its surface. There's a spike protein, there is a matrix protein, there is an envelope protein, and there is a hemagglutinin protein. So what these people did, that the spike protein is the largest one, and they took a sequence which they think that this could be a good antigen, and they produced an mRNA from that particular sequence, and they packaged those mRNAs into nanoparticles. Mainly these are liposomes, and these were injected into the humans. And then once these liposomes get inside the cell, these mRNAs will be released and produce that particular peptide or the part of the protein of the viral spike protein. And your immune system automatically will raise antibodies against that. It will be identified as a foreign reagent or a foreign antigen. And that's the route for the mRNA vaccine. But there are many other different routes by which the reverse vaccinology is being developed, where all the sequences of the SARS coronavirus surface proteins, all the 4 proteins that we talked about, the spike protein, the envelope protein, the hemagglutinin, and the matrix proteins are completely known. And the scientists are trying to find that which could be a proper antigen and which could be used over the others. There are many other different routes also, attenuated virus being developed, a full subunit vaccine also being developed. These are all at the preclinical stage, and at this point of time, it will take about a year or a year and a half before the vaccine comes out. If you're interested, you can just look at that where it stands. There is a website called clinicaltrial.gov. Again, clinicaltrial.gov. You can go there and look at that what SARS coronavirus 2 vaccines are at the clinical trial or preclinical trial stage. Perfect. So next we are talking about the biofuel production. It's also a very dynamic field because think about that. This is a huge issue worldwide because of several reasons. That think about that 2050, the population is expected to increase by at least 1.8 billion people, reaching about 9 billion worldwide. And the population economic growth also need increased energy demand. But unfortunately, what happened, the fossil fuels do have finite resources and contributors to global greenhouse gas elevations. So basically, what all the different nations in the world, they try to keep the carbon dioxide level to about 450 ppm by the turn of the century. So to do that, what you have to do that you have to find some alternative fuel. And you all know that electrical vehicles and those are being already made. But there are some other like ethanol and hydrogen. And the US Department of Energy set a goal by substituting approximately about 30% of gasoline with biofuels by 2030. So what does this mean? So ethanol can be used directly as a biofuel or added to the existing petroleum-based fuel. So you already know that there are many cars which approximately take 10% of our gasoline is added with the ethanol. And there are some E85 vehicles which runs on ethanol as well. But again, that's not the mainstream, not all the cars. Now, let's take a look at how we produce this ethanol and what are the challenges. So the ethanol can be produced from the degradation of plant starches in the corn. So corn is a source which contains these amylases and amyloglucosidases. So this is followed by the microbial fermentation of the sugars. But there are some limitations. As you are growing corn, you also increase the pesticide and fertilizer use and drives up the production of food prices as well because corn is a food product as well. So what's the alternative? So the alternative is that you can use a crop residue or waste material to ferment cellulose, and the hemicellulose is beginning to replace the corn-based biofuel production. Now, when you break down the cellulose and the hemicellulose, you produce 5 different types of sugars. So that is glucose, xylose, mannose, galactose, and arabinose. And to break down this cellulose and hemicellulose, what you need to heat these in acidic condition at about 100 degrees Celsius. That will break down this cellulose and hemicellulose into these sugars, which can be utilized for fermentation. But the problem is that, first of all, it's a very harsh condition. And then there are some microbes which has been now used to break down these cellulose and hemicellulose into the sugars. But the problem is that you can only use the glucose out of that to ferment, and the microorganism will ferment the glucose to produce the ethanol, but they will not use the xylose, mannose, galactose, or arabinose, or they will not ferment those other sugars. So there is a research underway to manipulate or find microorganism which can also use the 4 other sugars. Remember that the cellulose have 5 different sugars upon broken down. So only the 1/5 of the sugars that is being produced, which is only glucose, is usable to make ethanol. 4 others are not. So the scientists are trying so that the other 4 sugars are also possible to convert by fermentation into ethanol, and that will reduce the cost significantly. And that's a huge part of the research which is undergoing. But there is a problem with the ethanol. Ethanol do absorb water, and it can't be shipped through existing pipeline because, again, it must be distilled 2 times to remove the water. So it must be distilled at the source. Then if it is sent by a pipeline, it's going to, by condensation, is just going to get water in there. And then you have to distill it once again at the place where it is being delivered. So these are some of the problems. Also, it has less energy in its bond than the other molecules, including the gasoline and the butanol as well. So it takes more energy input to produce and refine it than you by using over the gasoline. So it isn't carbon neutral. So that is the problem. So it is really not helping that much. So because of the drawbacks, this is an area of growth and tremendous potential for innovation and growth. And we're going to take a look at what are the other things that can be done. So what about the hydrogen? So hydrogen does have 3 times more potential energy per unit weight than gasoline, and it is the highest energy content fuel available, surprisingly. And the problem is that storage distribution, it cannot be mixed with gasoline like ethanol to reduce petroleum usage. And if we wanted to use it, where could we obtain large amounts for? So you can find some of the fuel cell vehicles or hydrogen-driven cars that are available, but they are still not mass-produced, and many of these automobile industries are actively working along with the electric vehicles to hydrogen-powered vehicles as well. So hydrogen is produced as a direct product of fermentation, and is an oxygenic photosynthesis, and oxygenic photoheterotrophs also produce the hydrogen gas. And what happens is that the hydrogenase and nitrogenase enzymes both produce hydrogen gas. But the problem is that the process is very sensitive to oxygen. And the anaerobic microbes are also very difficult to deal and grow. So these are some of the drawbacks of the industrial hydrogen production, where you have to use these anaerobes to grow under anaerobic condition to produce these hydrogens. So there are also current efforts to make the process cleaner and more efficient. So let's take a look at the growing of the microbes in an industrial setting, and it does have its challenges. So one of the things that, before going into the material that we're going to talk about, so term fermenter. So what we mean by fermentation or a fermenter. So the fermentation, if physiologically or biochemically, if we are defining that particular term, what we mean by the fermentation is that when you have this pyruvate or other organic molecule to reoxidize this NADH to NAD+, and you produce an energy-rich product, say lactic acid or ethanol, that process is known as fermentation. But in industrial microbiology, that is not the case. What you do that in industrial microbiology, a fermentation means growing a microorganism or plant cells or animal cells in a large amount. So when you grow something in a large amount in an industrial setting, you call this fermentation, very generalized term. And the vessel that you use to grow those microorganisms is called a fermenter. So that's basically the term that is used in the industry. So what it needs to grow these different types of microorganisms, you need a precise control of agitation, temperature, pH, oxygenation. And this is basically the mass culture of microorganisms that I just explained. And then you scale up to a large fermenters. What do you mean by the large fermenter? Some of the fermenters can be 2 or 3 liters. Some of them could be 100,000 liters as well. And some of them can be 1,000,000 liter. One of the largest fermenter in the world, you can just Google, it is in Billingham in UK. And this fermenter is 200 feet high and 25 feet in diameter. And it is used for the flocculent yeast to ferment. And this is an airlift fermenter. What we mean by that from the bottom, the air is constantly purged, and that mixes the whole material. And it is a very homogeneous condition to brew. So it's an alcohol fermentation. In that 200 feet high fermenter, the mixing takes just about 10 seconds. So it is homogeneous, and it is amazingly efficient that how these engineering processes can be done. And along with that, you have to modification of the growth processes are necessary so that it can work efficiently in large fermenters. So here, on the other hand, we are looking at a stirred fermenter. So this is a continuous feed fermenter, and these can be in size of 2-3 liters to about 100,000 liter. But again, beyond 100,000 liter, these stirred fermenters actually do not work because there is too much of heat produced and there is not much efficient cooling. So the previous fermenter that I was talking about, the largest fermenter, which is a 1,000,000 liter plus fermenter, that is an airlift fermenter. There is no impellers inside. So mixing doesn't happen with the impellers. Instead, there is air purged from the bottom, and this air bubbles through the fermenter, and that is called an airlift fermenter without any impeller. So what happens that this unit can run under oxic or anoxic condition, and the nutrient addition, sampling, fermentation monitoring can be carried out under aseptic condition. And most of these things are completely automated. And there are biosensors, and infrared monitoring can provide a real-time information on the course of the fermentation, and then specific substrates, metabolic intermediates, and the final products can also be detected automatically. And also there are some foam control agents added if there is a protein-rich media, so that there is not much foaming in there. On the other hand, in this condition, when you're doing the fermentation, the condition changes over the period of time. So depending upon what kind of fermentation you are doing, there are sometimes batch fed or there is a continuous feed. So what is important that sometimes if glucose is one of the substrate, you don't want to add too much amount of glucose right at the beginning when there is not enough growth of the microorganism, because what it will do, it will produce a lot of these volatile compounds and your amount will diminish. As your microorganism is growing over the time, you add the nutrients accordingly to maintain a constant condition. Continuous feed or batch feed, depending upon what you're trying to do. But these fermenters also can act as what is called as a chemostat. So the chemostat is where you keep the growth of the microorganism constantly at the log phase. So in that condition, what you do, that you continuously feed a certain amount of culture and nutrients into the media, and you harvest the material accordingly as well to keep the log phase in there. But all fermentation processes are not like that. It does have an initial lag phase, log phase, and a saturation phase, at which step the fermentation is stopped, all the material is harvested, the whole thing is cleaned and sterilized, and a fresh batch is started back again. So that is called a batch fermentation compared to a chemostat or constant fermentation. So there are all different types of a variation in industrial fermentation. So also another very important thing that to know that when your microbe is producing what you want is critical. So the primary metabolites are the products of the microbes related to synthesis of microbial cell in the growth phase. And these include your amino acids, nucleotides, fermentation end products like ethanol and enzymes. On the other hand, your secondary metabolites actually accumulate following active growth. And many times this is considered as a part of a stress response where the nutrients are being depleted. We saw that in the very first slide in the antibiotic production. And antibiotic production is in this category. So that's pretty much all of your industrial microbiology and biotechnology. There are many different ways to manipulate the microorganism, but that is discussed in somewhere else in a different course. So basically what we do, that there are many different problems exist in the world around us and possibly into the future, and how cleanly using the microorganism we can produce these rather than doing harsh chemical reactions, and more efficiently using the microorganism. That is basically the goal of industrial microbiology and biotechnology.